Welcome back to the Deep Dive. Today we're tackling, well, a really fundamental question in biology. It's about how traits actually get passed down, generation after generation, without just blending together.
Right, before the 1860s, that was the prevailing idea, wasn't it? Like mixing paint, tall parent, short parent, you get a medium kid. Seems logical, but
But completely wrong, as it turned out. Thanks to Gregor Mendel, working away quietly in a monastery garden, he really turned that whole idea on its head.
he absolutely did. And that leap, realizing inheritance involves discrete units, not fluids. That's why people put Mendel up there with, like, Copernicus and Darwin. It was a genuine scientific revolution.
So our mission today is a deep dive into Chapter 3 of the text, Mendelism, the basic principles of inheritance. We're aiming to give you a quick, solid grasp of the core laws. Think of it as a shortcut to understanding classical genetics.
And the key really was Mendel's method. His genius wasn't just the idea, it was how he tested it. Choosing the garden pea, Pisum sativum, was, well, brilliant.
Why peas,
Right.
specifically? What was so special about them?
Well, a few things. They grow fast, they're easy to cultivate, and crucially, their flowers naturally self-fertilize. This meant he could easily get true breeding strains.
True breeding meaning they always produce offspring like themselves?
Exactly. If you let a true breeding tall plant self-pollinate, you always get tall plants. Same for dwarf plants. That gave him a reliable starting point. And, maybe most importantly, he didn't try to track everything at once. He focused, just one or two contrasting traits at a time, like tall versus dwarf.
Okay, let's walk through that classic experiment then. The monohybrid cross-focusing just on height. He took true breeding tall plants, the pea generation,
And crossed them with true breeding dwarf plants. Simple enough start.
and the result, the F1 generation.
All tall, every single one. The dwarf characteristic just seemed to disappear completely, which right away contradicts that blending idea. You'd expect medium height.
Okay, wait. If it disappeared, how did he know the dwarf factor wasn't just gone, like destroyed somehow? How did he know it was still there, just hidden?
Ah, that's the critical next step. He took those tall F1 hybrids and let them self-fertilize. This produced the next generation, the F2.
And if the dwarf factor was really gone?
It couldn't possibly come back,
Okay.
but it did. He got both tall and dwarf plants in the F2 generation, and not just randomly, but in a really consistent pattern. His actual numbers were 787 tall and 277 dwarf.
Which is almost exactly.
3-to-1 ratio. That consistent mathematical relationship was the evidence. It suggested something particulate, something quantifiable was being passed down.
Right. So let's nail down the terminology here. These heritable factors are what we now call genes. And the different versions, like the factor for tallness versus the factor for dwarfness, are alleles.
Correct. And we need to distinguish between the genotype, that's the actual combination of alleles an individual has, say, using D for tall and D for dwarf, could be DD, DD, or DD.
And the phenotype, which is just what it looks like physically. So tall or dwarf.
Exactly. And this F1, F2 result leads directly to Mendel's first big idea, the principle of dominance. Basically, one allele, the dominant one, we read it uppercase D, masks the effect of the other allele, the recessive one, lowercase d, when they're paired together in a heterozygote, which is DD.
So the D to plants look tall because D is dominant over D. Makes sense. But what about that 3.1 ratio? How did the dwarf trait reappear? That's the second principle, right?
Yes, the principle of segregation. This is really the core rule of how traits are transmitted. Mendel figured out that the two alleles an individual has for a trait must separate or segregate when they make the reproductive cells, the gametes, sperm, and eggs.
Ah, so this happens during meiosis.
Precisely. Meiosis ensures that if a parent plant is DD, its gametes don't carry both D and D. Half the gametes get the D allele and the other half get the D allele. You go from diploid, having 2 copies, to haploid, having just 1 in the gamete.
And when fertilization happens, these gametes combine randomly.
Restoring the diploid state in the zygote. And it's that separation and random recombination that mathematically generates the 3.1 phenotypic ratio you see in the F2 generation. It's not magic, it's probability based on segregation.
Okay, so segregation covers one trait, but obviously organisms have tons of traits. How did Mendel tackle that? Did he look at multiple traits at once?
He did. That led to his dihybrid crosses, where he tracked 2 different traits simultaneously. The classic example is seed shape and seed color.
Right, remember this one. Yellow and round seeds versus green and wrinkled seeds.
Yep. He started with true breeding parents. One was yellow and round, let's say genotype GGWW, both dominant, and the other was green and wrinkled, GWW, both recessive.
And the F1 generation, I'm guessing, showed only the dominant traits.
You got it. All F1 plants, genotype GWW, produce seeds that were yellow and round. Dominance holds for both traits.
But the real test comes in the F2 when you cross those F1s.
Exactly. And what he found was fascinating. He didn't just get the original parental combinations back, yellow round and green wrinkled. He also got new combinations, yellow wrinkled and green round.
Ah, so the traits weren't inherited as a package deal.
Precisely. They appeared independently. And the numbers fell into that famous ratio. 9 yellow rounds, 3 green round, 3 yellow wrinkled, and 1 green wrinkled. The classic 9.3.3.1 phenotypic ratio.
And that specific ratio is the evidence for.
The principle of independent assortment. It means that the alleles for one gene, like seed color, GE, segregate into gametes independently of the alleles for another gene, like seed shape, WW. It's like shuffling two separate decks of cards. What you get from one deck doesn't influence what you get from the other.
And biologically, that's because these genes are typically on different chromosomes, right?
Yeah.
Or far apart on the same one.
That's the physical basis, yes. The way different chromosome pairs line up and separate during meiosis is independent, which leads to the independent assortment of the genes they carry.
Okay, so we have the three big principles: dominance, segregation, independent assortment. Now, how do we actually use these principles to predict the outcomes of crosses? This seems like where it gets practical.
It does, and there are a few tools geneticists use. For simpler crosses, maybe 1 or 2 genes, the Punnett square method is really useful. It's visual, systematic. It helps you list out all possible gamete combinations from each parent and see all the potential zygote genotypes.
Like mapping out those 16 possibilities in the dihybrid F2 cross.
Exactly. But as you can imagine, if you add a third trait, a trihybrid cross, well, a Punnett square gets unwieldy. Fast. You'd need 64 squares.
Yeah, sounds like a recipe for mistakes. So what's the alternative for more complex crosses?
The fork line method, also called branching diagrams, it's much more efficient. You basically treat the multi-gene cross as several independent single-gene crosses happening at the same time.
How does that work?
You take the expected ratio for each gene separately, like for a heterozygote cross, it's 3 dominant, 1 recessive phenotype. Then you use branching lines to combine these ratios. For a trihybrid cross, like DDGGWWX, DDGWW, you'd combine 3 separate 3.1 ratios to get the overall phenotypic ratio.
Which comes out to that monster 27.9.3.3.1 ratio, right?
Yeah.
Much faster than drawing 64 boxes.
Much faster. And arguably the quickest method, especially if you only need the probability of one specific genotype or phenotype, is the probability method.
Using math rules.
Yeah, basic probability. The key is the multiplicative rule for independent events. If you want the chance of getting, say, a fully recessive offspring, AABBCC, from a trihybrid cross of heterozygotes, ABBCC, XABBCC, you just multiply the individual probability.
Okay, so the chance of getting A is 14, BB is 14, CC's is 14.
Right, so you multiply 14x14, x14, x14 is 64. Super quick. And if you want the probability of one outcome or another mutually exclusive outcome, you use the additive rule. You just add their probabilities together.
Got it. Now, before we get to testing hypotheses, there's one more practical tool mentioned, the test cross. What's that for?
The test cross is super useful. Let's say you have a plant showing a dominant phenotype, like a tall pea plant. You don't know if its genotype is homozygous dominant, DD, or heterozygous, DD. They look the same, right?
Yeah.
To figure it out, you cross that unknown tall plant with a plant you know is homozygous recessive, DD, the dwarf plant. That's the test cross.
And how do the results tell you the unknown genotype?
Because the recessive parent can only contribute D alleles. So if any of the offspring are dwarfs, EDD, it means the unknown tall parent must have contributed a D allele. Therefore, the unknown parent had to be heterozygous. If all the offspring are tall, the unknown parent was likely homozygous dominant. DDD directly reveals the alleles carried by the unknown parent.
Clever. Okay, so we have ways to predict outcomes. But experiments don't always match predictions perfectly, right? There's random chance involved. How do we know if our results are close enough to what Mendel's laws predict?
Excellent question. That's where statistics come in. We need an objective way to decide if the difference between what we observed in an experiment and what we expected based on our hypothesis, like a 3.1 or 9.3.3.1 ratio, is just due to random fluctuations, or if it's so large that maybe our hypothesis is wrong.
And the tool for that is the chi-square test.
Exactly. The chi-square test, written as chi to 2. It gives us a single number that summarizes how well the observed data fits the expected data.
How's it calculated generally?
The formula looks a bit scary, but the idea is simple. For each possible outcome category, like tall plants or dwarf plants, you calculate the difference between the observed number, O, and the expected number, E. You square that difference and then divide by the expected number, E2. You do this for all categories and then sum them up. That total is your chi-2 value.
Okay. And a low chi-square value means?
A low chi-2 means your observed numbers are very close to what you expected. Good fit. A high chi-2 means there's a big discrepancy. Bad fit.
The source material gives a great comparison here. Mendel's own dihybrid cross data, when analyzed, apparently gave a really low chi-square value.
Yeah, remarkably low, something like 0.51, which means his results were incredibly consistent with the 9.3.3.1 ratio predicted by independent assortment. Almost too good, some have joked, but definitely supportive.
But then it contrasts that with data from Hugo de Vries, one of the scientists who rediscovered Mendel's work.
Right, de Vries did a similar two-factor cross with different plants, campions, and his results, when put through the chi-square test, gave a value of 22.94. Much, much higher.
So how do you decide if a chi-square value like 22.94 is too high? Is there a cutoff?
Yes, there is. You compare your calculated chi-square value to a critical value found in a statistical table. To find the right critical value, you need to know the degrees of freedom for your experiment.
Degrees of freedom. That's related to the number of categories.
Usually it's the number of outcome categories minus 1. So for that dihybrid F2 cross, there are 4 phenotypic categories, 9.3.3.1. That gives 4.1 was 3 degrees of freedom.
Okay, so for 3 degrees of freedom, the text mentions a critical value of 7.815.
Right. That value corresponds to a standard scientific threshold, usually a 5% probability level, P05. If your calculated 2/2 is larger than the critical value, like de Vries' 22.94 is much larger than 7.815, it means the deviation you observed is statistically significant.
Meaning it's unlikely to be just random chance.
Exactly. There's less than a 5% probability you'd see such a large deviation if your original hypothesis, independent assortment in this case, was actually true. So objectively, de Vries should have questioned whether independent assortment applied to his campion traits, even if he thought his results generally supported Mendel. It shows why having that objective statistical test is crucial.
Makes sense. Okay, we got the principles, the prediction tools, the statistical test. How does all this apply when we can't do controlled crosses, like in human genetics?
Yeah, studying human inheritance is obviously trickier. You can't tell people who to marry, and family sizes are usually small compared to Mendel's hundreds of pea plants. So we rely heavily on analyzing existing family histories using pedigrees.
Those family tree diagrams with squares and circles.
That's them. Squares represent males, circles represent females. Usually a shaded symbol means the individual is affected by the trait being studied. By looking at how the trait appears across generations in a pedigree, we can often deduce its mode of inheritance.
So what patterns do you look for to tell if a trait is, say, dominant versus recessive?
Well, for dominant traits like achondroplasia, a form of dwarfism, or Huntington's disease, you typically see the trait appearing in every generation. Affected individuals almost always have at least one affected parent. It doesn't usually skip generations unless it's a very rare new mutation.
Okay, and recessive traits.
Recessive traits like albinism or cystic fibrosis can skip generations. You'll often see unaffected parents having affected children. This happens because the parents can be carriers heterozygous carrying the recessive allele without showing the trait themselves.
And these are more likely to pop up if relatives have children together.
Yes, that's a key indicator for rare recessive traits. Because relatives share a higher proportion of their alleles by descent, there's an increased chance that both parents might carry the same rare recessive allele, leading to an affected child. First cousins, for example, share about 1/18 of their alleles.
Wow. So pedigrees help us figure out the inheritance pattern. How does this translate into practical advice, like in genetic counseling?
Genetic counselors use Mendel's principles, especially probability, all the time to assess risks for families. But they have to be careful because, as we said, human families are small. The expected ratios are just probabilities, not certainties.
You mean like even if 2 carriers for a recessive condition, say CCXCC, have kids, they might not get exactly 3 unaffected and 1 affected.
Right. That 3.1 ratio is the expected outcome over many, many offspring. For a single family with, say, 4 children, the most probable outcome might be 3 unaffected and 1 affected, but other outcomes are definitely possible. The text mentions using binomial probability to calculate the chance of specific combinations in small samples. The chance of exactly 3 unaffected and 1 affected is actually 108 out of 256 possibilities.
So it's about likelihoods, not guarantees.
Exactly. And counselors use this to calculate individual risks too. For instance, consider someone healthy, let's call them R, whose parents are known carriers AAXAA for a recessive condition. What's the chance R is also a carrier AA?
Hmm. Well, the possible genotypes from AXAA are AA, AA, and AA, which is the same, and AA. R is healthy, so they can't be A. That leaves AA, AA, and AA.
Perfect. So out of the three possibilities compatible with being healthy, two of them are the carrier state, AA. Therefore, R has a 23 chance of being a carrier. That 23 risk is a really common and important calculation in genetic counseling.
That's a really clear application. So let's recap this whirlwind tour of Mendelism. We've covered the foundational principles.
Dominance, where one allele masks another. Segregation, how alleles separate during gamete formation. And independent assortment, how different genes are typically inherited independently.
And we looked at the toolkit. Punnett squares for visualizing crosses, the forked line and probability methods for handling more complexity, the test cross for finding unknown genotypes.
And crucially, the chi-square test for objectively evaluating how well our experimental data fits the predictions from these principles.
These ideas and tools, developed over 150 years ago from studying peas, are still absolutely central to genetics today, aren't they?
Absolutely fundamental. They form the basis for predicting and understanding inheritance patterns in everything from plants to people. But, as the source material itself hints, that third principle, independent assortment, isn't always true.
Ah, so there's a twist.
There is. While Mendel's laws provide the essential framework, the idea that all genes assort independently has exceptions. Which leads us to the final thought for you to ponder.
Go on.
What biological mechanism operating right there inside the cell nucleus during meiosis could actually cause the alleles of two different genes to not assort independently? What could physically link them together, making them defy Mendel's third law? We'll leave that thought hanging for now, perhaps for a future deep dive.